It's my pleasure to introduce Dr. Marc Branch for this afternoon's address. Probably Marc doesn't need much of an introduction. He's published over 80 scientific articles in the field of behavioral pharmacology. Served on several. I can talk about it.
How many articles in the field?
Served on several editorial boards and NIH study sections. But I didn't think that would stop me, so I thought I'd give him an introduction anyway. Forward. Outline of my introduction. Since Marc's going to talk to us a little bit about the history of behavioral pharmacology, I thought I'd talk about a little bit about Marc's history. So my outline is where Marc came from, what Marc has done, a Marc Branch story, and if there's any time remaining, Marc will give his tutorial. Marc's history. Marc probably doesn't need to use notes either because, as you can see, he's from the Harvard line, trained at the University of Maryland in Lou Golub's lab, and trained Skinner at Harvard. Next slide shows some of Marc's major accomplishments. These are all the PhD students that Marc has trained. What you can't see are the master's students in there, and current list of students on the right. So I guess there are about a dozen of us in this room right now, I think. So we consider that some of Marc's major accomplishments. We hope he does too.
Yes, he does.
Let's see, the story. I'm almost finished. This is the second Marc Branch story in my repertoire. Learned this story about 24 years ago when I first started my graduate work at the University of Florida. And I walked in upon a bunch of students studying for a behavioral pharmacology test. And they were studying pretty hard. They've been doing it for days. And wondering what I got myself into, I asked them, what were Marc's tests like? How hard are they? And Randy Scruggs told me the story that, well, let me give you an analogy. It's like studying for a test on the alphabet. So you study pretty hard and learn all the letters, you learn the history of the alphabet. You can do it backwards, you can do it forward. You can skip every third letter going forwards, every fourth letter going backwards. You get ready to take the test, you walk in. And the first question is, what three letters are needed? The second question, if you can add a letter, what would it be? So it is my pleasure to introduce Marc, knowing that we'll get to ask the questions after this tutorial.
Well, I'm relieved. When Steve had a rack of slides, I thought maybe something scandalous might appear, because he's got slides of that too. Having been but luckily that didn't happen. Actually, I'm not sure which way the tutorial is going to go here today. As I look out in the audience, I see people who know probably more than I do about behavioral pharmacology and what it is, so I'm not sure why those people are here. I also see a coterie of students who just finished my course in behavioral pharmacology who are here. I'm not sure what that means. Maybe they didn't get it. But let me begin. As some people, actually we have some students from Florida here, know that one of the things that I occasionally do in oral exams on theses and dissertations is have people defend the title just to get them off to a good start. And so I probably have to defend this one a little bit because it was written in haste and there are a couple things about it I don't like already. Now the behavioral pharmacology part I'm comfortable with. The what is it is presumptuous in at least two ways. One, it suggests that there's an it, some sort of platonic essence to behavioral pharmacology, which I'm quite sure there's not. Behavioral pharmacology is a subfield within psychopharmacology. I mean, it's a pretty small, but even though it's circumscribed, it's got great variety, and it's subsumed essentially by what people who choose to call themselves behavioral pharmacologists actually do. So there's no it. And even if there was an it, who am I to tell you what it is? That's the other presumptuous side of the arrangement. But it does afford me the opportunity to tell you what I think it ought to be, which is sort of what you're going to hear here today. So I'll give you a quick summary here, sort of a preamble. These are the topics I'm going to try to cover. I might not get to the eighth one. I was feeling a little embarrassed. I went to a talk a couple hours ago by Tom Critchfield, in which he also had 8 points, but he also had 28 subpoints, which is pretty impressive. I don't have, and this was only a 30-minute talk, so I don't know how I'm going to do here. I'm going to talk about an important early experiment. Then I'm going to talk about a logical basis for sort of defining behavioral pharmacology, what it's about. I'm going to talk about some parallels between behavior and analytic techniques and those used in pharmacology. Then I'm going to have the tutorial part, quick tutorial on dose-response functions, a little bit about my views of what's meant by mechanism of action, especially a behavioral mechanism of action. Then some examples of ways to illustrate the importance of behavioral factors in effects of drugs. And also, if I get a chance, to talk about how experiments with drugs might tell us something about behavioral processes. And finally, if there's any time left over, I'll talk about what's called behavioral pharmacology, at least as I see it, is these days. So let me begin by talking about an early experiment. And this is an experiment which I think shows a discovery that still astounds me. And I usually point to it as the experimental report that started what I view as behavioral pharmacology. Interest in drug effects on behavior has percolated around for centuries. And the advent of the drug chlorpromazine in the early 1950s for the treatment of psychosis spurred interest in drug effects on behavior. But there's shortly in the middle 1950s, Peter Dews conducted a very simple experiment which yielded what I find a very interesting result. Dews was a pharmacologist at Harvard who knew about Skinner's lab, went over and found some interesting things about it that we'll talk about in a few minutes, and thought this was an interesting place maybe to do some experiments, examine the effects of drugs on behavior. So the experiment involved food-deprived pigeons. Key pecking in the ordinary way was established under either an FR50 or an FI 15-minute schedule of food presentation. And once performance had become relatively stable, some sessions were preceded by injections of sodium pentobarbital. Pretty simple, straightforward experiment, essentially comparing two different schedules of reinforcement. Well, what happened? Well, some interesting things. This is a recreation of the main figure from this experiment. Actually, can I put it a little higher? Some people in the back seem to be craning necks. By the way, since this is allegedly a tutorial, you're supposed to be allowed to interrupt, and I'm happy to take questions. If I make anything that's really confusing, don't ask. What this shows effects on response rate of a range of doses of sodium pentobarbital, expressed as absolute doses, actually, from 0.25 to 5.6 milligrams per pigeon. And the effects are expressed as a proportion of control rate with rate after the drug divided by rate without drug. So it's just a proportional measure. The leftmost dots here show the effects of injecting the saline solution vehicle. That's what the drug was mixed up in. These drugs were injected into the breast muscle of the pigeons. And these each show the effects of a single dose, which comes from a single test period. These test periods are spaced by a few days so that performance can recover. And so the fixed interval 15-minute, take a look at those data first. The smaller doses seem to bump it up, at least as compared to saline, maybe not as compared to control. And then larger doses decrease it, illustrating the importance of dose, that the drug isn't simply a depressant or a stimulant, depends on the dose. That's an important message. But the more dramatic message here is the difference between drug effects between the two schedules of reinforcement. That under, especially dramatic at some of these larger doses, that at 1, 2, and 4 milligrams of pentobarbital, response rates are essentially unaffected or maybe a little above control level under FR50. They're almost eliminated under FI 15. The way I like to describe this is what we have here is we have a subject doing the same thing, pecking a key for the same reason, to get food, yet the drug effects can be qualitatively different, either a stimulant or a depressant effect at a particular dose, depending on the schedule of reinforcement. It's a remarkable discovery. If you're not astounded by that, I suggest you leave, because everything else is built on being astounded by this. I still find this astounding. It's an amazing effect. And the effect, I can sort of segue that, I hope, into a logical description of some sort of quasi-logical description of where behavioral pharmacology comes from, how it emerged. It's a little cheap syllogism. Syllogism that helps define the field, as it says here. The major premise is that different kinds of behavior may be affected differently by a drug. That's an empirical fact. I just showed you a slide illustrating that effect. There are many, many illustrations of that, even before Dews's work. For example, there was evidence before that chlorpromazine, the drug that a lot of people point to as the start of real psychopharmacology, has differential effects on avoidance and escape behavior. That avoidance behavior is more sensitive to its effects than is escape performance. So there's lots of evidence for that. Second premise, different kinds of behavior can be produced by different histories of experience. I point to experimental psychology in general, or perhaps all the issues of SQAB to illustrate that point. That's also an empirical fact. So the conclusion is, maybe it's the case that different histories, for example, exposure to contingencies, can result in different drug effects. And in fact, that's what Dews's experiment showed. So if that's the case, then there's something to be explained there, and that's behavioral pharmacology's business. Build a theory of how behavioral variables, like contingencies of reinforcement, determine behavioral effects of drugs. So that's sort of the tack on behavioral pharmacology that I like to take and that I like to sell. Now, behavior analysis, or the experimental analysis, is certainly one part of the science, my favorite part of the science, about how experience determines current behavior. So it seems like a good place to start to try to examine the effects of drugs on behavior, so that we can take pharmacology and the experimental analysis of behavior, put them together, and maybe we've got something where we can learn something about drug effects on behavior. Now, one of the other things that made it convenient and useful, I think, for behavior analysis and pharmacology to merge in this way are there are some common characteristics in the way basic pharmacology is done and the way basic research in the experimental analysis of behavior has been done. And there is that parallel can be illustrated in the concept of a pharmacological preparation, talking about some of the parallels between experiments in behavior analysis and experiments in pharmacology. Usually when people talk about doing drug experiments, they talk about having a preparation to which they add drugs. In the case, and there are many parallels between basic pharmacology and the experimental analysis of behavior, the procedures that are widely employed. There are many parallels here. The first I'll point to is that in basic pharmacology, a very common technique is to take an isolated piece of tissue, usually an esoteric piece of tissue, like a guinea pig ileum or a frog diaphragm. The rest of the frog is gone. You know, guinea pig, the rest of the guinea pig is gone. Rat testis, little pieces. In a similar way, you can think that the experimental analysis behavior has chosen as an experimental tactic to isolate little pieces of behavior. You have to remember that's what you're doing, as Charlie Shimp pointed out to us yesterday, that it's not the whole world. It's a little teeny piece of it to try to get a clearer picture. So we isolate pieces of tissue. No one complains, by the way, when physiologists do this. You know, when somebody figures out a way to isolate a piece of tissue to study it, it's a great achievement. It's not criticized because it's ignoring all the other variables, or it isn't real life. So there's certainly that kind of similarity. In both cases, a controlled environment is provided for the thing to be watched, the piece of tissue or the isolated bit of behavior. In the case of pharmacological preparations, it's usually some sort of bath that mimics body fluid, has the right nutrients, has the right amount of oxygen, has all the other things that help keep that piece of tissue about as alive as it would be if it were still inside the organism, except it isn't, so you can watch it better. Same way in operant conditioning still represents, I think, I'm not sure we're at the ultimate, but it's certainly a vast advance over other kinds of techniques where extraneous variables are not excluded to the extent that they are in operant conditioning chambers. That hardly means that they're all gone in operant conditioning chambers. As all of you who have ever done experiments in operant conditioning know, there are lots of things in there that we wish we could get out. I keep waiting for bright young students to invent new and better techniques of isolation, because I think at some point you do have to, as Peter said earlier this morning, you have to step back when you have to do that and see where you are in the big picture. But it's a lot easier to see things in experiments in the little pieces. Both typically involve, or at least frequently involve, a baseline. In the pharmacological preparation, it's often the contraction of the piece of tissue. It's suspended. Automatic measurement of the contraction is arranged. And you wait, put it in the bath, and you wait for that contraction, which is variable. You know, while it's alive, it's contracting a little bit, up and down. And same thing with the behavioral baseline. Boom, 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 boom. We usually do this over successive sessions. So one of the big differences between pharmacology and behavioral pharmacology is how fast individual drug experiments get done. You can do a whole set of experiments in one afternoon in pharmacology. The other advantage to the basic pharmacological procedures is that after you add drug to the preparation, there are ways to get it off there. You can wash by doing repeated washings of the tissue, you can actually eventually get the drug off and have your preparation ready to go again. And that's how you can do a whole experiment in an afternoon. You have to wait for natural metabolic processes to get rid of drugs in whole animals, which is what behavioral pharmacologists study. It's possible to test the effects of the vehicle. It's important to test the effect of whatever you're mixing the drug up in. Typically, drugs that you get, they come as powders, a little purer than you can get on the street corner, that you can mix to prescribed concentrations, and usually you pour in. And some drugs, actually, they won't dissolve in inert kinds of vehicles, and so you need to test the effect of the vehicle itself. That can be done easily. You add it to the bath, or you add it to the organism. In Dew's experiment, he added it to the organism. Test each dose individually, and each dose test is really an ABA design. You've got baseline, you stick in the dose, see what happens to the dependent measure, their contraction or rate of response or percent choice or whatever it is you're looking at, and then wait for the baseline to come back. It's sort of a probe kind of arrangement. And in many cases, a single preparation is all you need. It's another parallel between pharmacology and the experimental analysis of behavior. It's always nice to have a second one just in case. But if you can get meaningful, interpretable, reliable kinds of results from single preparations, one of the things that illustrates is you probably have a better understanding of what's going on than if things are all over the place and you need lots of preparations to average them together. In pharmacology, I don't think you very often see averages of huge numbers of pieces of tissue. That just doesn't happen. They don't have to because they know what's going on. So those parallels exist. So that made for an easy marriage between pharmacology and the experimental analysis of behavior. Well, now comes the little short tutorial part, just in case you didn't know about dose-response functions. I'm going to talk a little bit about dose-response functions, how you look at them, how you interpret them. They're not very complicated. They're graphs, and most people in here are very familiar with graphs. Y-axis has some sort of dependent measure, contraction, responses per minute, percent choice, whatever it is. X-axis is where the dose is displayed. Most of the time that's on a log scale. There are actually formal reasons why that's useful. It turns out that we'll talk a little later about the concept of receptor, and actually the idea that there are such things as receptors predicted particular forms of the dose-response function, which would appear most clearly with log, semi-log axes, log x-axes. Dose-response functions can be quantile or graded. Most of the ones we talk about in behavioral pharmacology are graded. That is, the dependent variable is continuous. Quantile dose-response curves are all or none measures. One of the most well-known in pharmacology is the all or none measure of did you die or didn't you, to produce, and those are used to produce population estimates of lethal doses, those kinds of things. Dose-response functions, in addition, if they have particular shapes with logarithmic x-axes, can be used to make inferences about what the drug must be doing wherever it's doing it at the molecular level. But they are also used to define some parameters of drug effects, and we've got three kinds of things to talk about here. And the first are the concepts of potency and maximum effect of a drug. Here are some made-up data, always the best kind. They illustrate exactly what you would like them to illustrate most of the time if you're good at drawing them. Two kinds of dose-response functions here. The ones on the left are monotonic, as you might guess, and the ones on the right are bitonic. Frequently in behavioral experiments, which are supposedly illustrated by the data on the right, drug effects tend to be bitonic, that there are two parts to the dose-response curve, an upward part and a downward part. Not always. I'll show you some other curves that go the other way. The concept of potency simply refers to how much drug does it take to get the effect. And so the potency is illustrated by where left to right in these graphs the dose-response functions lie. So in this one, drug X here is the more potent drug, even though it doesn't produce as large a maximum effect. Those are the two parameters that are separable. So potency refers to how much of the drug it takes to get an effect. Maximum effect refers to maximum effect. Very tricky concept to get through. But now, with bitonic functions, there are two maximum effects. There's the maximum increasing effect and the maximum decreasing effect. So in characterizing drugs X and drugs Y here, drug X is more potent. Drug Y has a greater rate increasing effect. They have equal rate decreasing effects. So those are the concepts of potency and maximum effect. Dose-response curves are also used to characterize drug interactions. And there are two major kinds of drug interactions. Synergism. And synergism, here's our original drug X. And here's dose of X. We've got some dose-response function, an increasing ogival function. Also have it over here. We've got two kinds of synergism and antagonism here that some people distinguish. Synergism, so I add a single dose of drug Z, and then I test all these doses of drug X again. If the curve goes this way, synergism, right? Potency increase. If it goes that way, it's antagonism. This illustrates what's called a surmountable antagonism. That is, I can get the ideal effect of X, I just have to give more of it. So I can when Y is in there, it takes more X, but I can eventually get to any old effect of X. I can surmount the effect of Y by increasing the dose. Surmountable antagonisms have implications about where drugs are acting. Often, one would argue, if you saw that, they're acting at the same receptor. Here's a little more complicated situation where we have Z increases the potency of X, and also you wind up with a larger maximum effect. Some people call that potentiation rather than just a synergism. It's larger. And here the antagonism apparently is not surmountable. And that usually means it's not a simple receptor-site interaction. So you can learn some things about what's happening with the drugs just from the dose-response functions. Third characteristic here, you can describe the phenomena of tolerance and sensitization via dose-response functions. Here's the original effect of whatever this drug is. I should have put dose of X, because these are all dose-response functions for X. And this is what happens after prolonged exposure to X, maybe. Maybe we get more sensitive to it. Potency shift to the left. Sensitization, that is the technical expression for that. If this is more common, curve moves to the right after extended exposure to the drug. It's referred to as tolerance. And a perfect parallel shift here is referred to as surmountable tolerance. That we can get the maximum effect, we just have to go to a dose of 100 instead of a dose of 10 to get the maximum decreasing effect here. I probably should have brought a graph, and I didn't, showing that one of the reasons for doing dose-response functions, especially if they're bitonic, is you can confuse yourself about whether it's sensitization or tolerance if you look at only a single dose. Imagine a function that looks like this as an original function. And then imagine that you give this dose a whole bunch of times and its effect drops down here. What is that? Well, a lot of people would call that tolerance. Well, it depends. If the curve went over here, yeah, it is, but what if the curve went over here? It'd be at the same spot, and you would have incorrectly concluded you had tolerance when you in fact had an instance of sensitization. So dose response functions are useful ways to keep from making mistakes. Now, I've made mention of drug receptors, and those fall as a special feature under the more general notion of mechanisms of action of drug. This is a concept that's pretty well worked out in pharmacology, physiology, and tried to draw some sort of analogy between physiological mechanisms of action and what I'd like to refer to as behavioral mechanisms of action. The idea of a mechanism of action is that somehow a drug interacts and modifies the normal functioning and interacts with the normal processes of the organism. An example that may make sense here is the effects of drugs on blood pressure. You give a drug and blood pressure changes. Well, how might that have happened? Well, people know about the processes that control blood pressure. Heart rate, stroke volume, peripheral resistance, there are reflex responses, and people know what those are. And so you go in and you ask, well, what's the drug doing? Is it increasing peripheral resistance? And if you find that, that is a mechanism of action. And it doesn't do anything to anything else. Its mechanism of action is increasing high blood pressure by increasing peripheral resistance. That would be a reasonable kind of description of a mechanism of action. Or it increases heart rate, doesn't do anything else. In physiological action of drugs, the notion of mechanism of action now is that certainly at one level, virtually almost all drugs are thought to act at what are known as receptors. As Jack Maar once wonderfully said, the receptor is the rare hypothetical construct that made good. That's a really nice description. But it has made really good, and it shows the great strength that a really well-described hypothetical construct can have in organizing data. We don't have receptors in behavior. We're not that far along. We think we have some understanding of the variables that ordinarily influence behavior. This is another one of the reasons for an attraction between pharmacology and the experimental analysis in defining this field of behavioral pharmacology, is that we think we have some idea of the factors that are responsible for behavior. And we know what some of those are, and so we can look to see how the drug interacts with them in a sort of semi-oversimplified outline. I'm sure I've left out variables here that will offend several people in the audience, but that behavior is a function of a variety of kinds of variables that have been studied, and some of their effects have been characterized, some not so well, but antecedent variables that are now generally referred to as establishing operations, things that current stimulus variables, what's the modality, what if the drug influenced hearing and you had an auditory discrimination? Well, you could test that by training the discrimination in some other modality to see if that's the mechanism by which the drug is disrupting a discrimination. Is it because I can't hear it, or I make it hear it differently? It may interact with timing processes so that the duration of stimuli exert control in a different way. Their topography of the response may make a difference. These are all variables that we know that make a difference in what we see in behavioral data, so maybe they would have some interaction with the way a drug influences behavior. And the way one would do that the problem is it takes lots of experiments to work this out. In fact, one of the more interesting semi-disappointments is that that experiment by Dews that I showed you was published in 1955. That's the experiment that sort of started what I characterize as behavioral pharmacology. A thorough attempt at characterizing the behavioral mechanisms of action of that effect has yet to be completed. So it's a slow process. Here you go, this is a big dissertation out there for some student, where you could do all the experiments that you would need to do to try to figure out where that effect comes from. Is it rate of reinforcement? Does it have anything to do with the topography or the different topographies of effect? Is it just this? We know it's not just the schedule. People have chipped away at this. How specific is it to a particular kind of establishing operation? All kinds of issues. Some of which, if you went through the literature, you could eliminate some. Maybe half of these, but there's plenty left to do there. But this is the notion that, this mechanism of action idea has also been useful for understanding physiology, and we think it might also be interesting for understanding behavior, sort of a bootstrap operation. If you have a way of conceptualizing, for example, how blood pressure is controlled, and then you do a drug experiment, none of that stuff seems to work, then it gives you ideas that maybe you've missed something. Similarly, if you give a behavioral effect, you say, well, geez, I thought it was an effect on positive reinforcement. And then you discover, well, no, it's not that, it's something else. Then you're learning something about the way behavior is controlled. And I'll talk a little bit more about that later if I don't trip and kill myself in the meantime. So now talk about a few ways of, at least, behavioral pharmacology at this point is full of illustrations of very interesting effects, very few of which have been analyzed from any perspective, let alone a behavioral mechanisms of action. But there are lots of illustrations about how behavioral factors modify the effects of drugs. And as in a book I read a long time ago, a little book by John Harvey, one of the first textbooks in behavioral pharmacology, he pointed out that one of the great challenges to pharmacology in general is predicting of individual differences in drug response. And it's clear that these kinds of variables, especially when studied in individuals, you can show huge variation in drug response depending on what seem like reasonably subtle factors. Now, certainly, the dependence of drug effects on schedule of reinforcement. Well, the first experiment that I show you here, Dew's discovery, illustrates that. It can make a big difference. And one of the reasons, actually, I think that the Dew's original effect hasn't been subjected to complete analysis, or at least attempt at analysis, is that people thought they had it figured out for a while. That, in fact, that the issue, that what really made a difference wasn't really the schedule, it was the rate of responding that it produced. And the fixed ratio schedule produced this higher response rate, and the fixed interval schedule produced a lower average response rate. And son of a gun, there were other experiments that seemed to, hey, low response rates are, if I got a low response rate, I get one drug effect. I got a high response rate, I got a different drug effect. And there were some experiments illustrating not really prepotent schedule dependency that seemed to be quite closely correlated with rate of response. And probably the most famous of these is an experiment by Kelleher and Morse in 1964. And no tutorial on behavioral pharmacology would be complete without talking about this experiment, because it had tremendous influence. It's a really very clever experiment, too, and with beautiful data, as you'll see. What Kelleher and Morse did is had squirrel monkeys, and they trained them under fixed interval and fixed ratio schedules. Some of the monkeys were trained under a fixed interval. They were both trained under fixed interval 30 and fixed interval 10. So the numerical parameters of the schedules were the same. One group of monkeys was trained with food pellets as reinforcement, sort of the standard. The other group of monkeys was trained with an aversive control, or negative reinforcement procedure, which they called termination of a shock stimulus complex. In the fixed ratio portion, the stimulus would come on, and 30 seconds later, shocks would start to occur, unless 30 responses had already occurred. Boom, then there was a timeout period. So the shocks could be avoided by completing the fixed ratio in a fixed amount of time. In the fixed interval part of that, the fixed interval stimulus stayed on for 10 minutes, and then at 10 minutes and 3 seconds, shocks started coming every 3 seconds, electric shocks. That's 3 seconds after the end of the fixed interval, which means if you make a response between the end of the fixed interval and 3 seconds after the fixed interval, you don't get any shocks. And in fact, that's what the squirrel monkeys do. They learn to do that. What they produced is virtually indistinguishable performances, at least with, as we go. They show cumulative response records, and you can't tell whether it's food reinforcement or shock avoidance reinforcement. They're virtually identical. So the patterns of responding, so you got lower rates in the fixed intervals, but they have classic-looking fixed interval performance, which, as we all know now, is a long pause, maybe a little acceleration and then a constant rate, and fixed ratio performance, which is a very short pause and then very fast responding in both components. Then, they examine the effects of some drugs on these. So now they've equated what the behavior looks like, but they have very different reasons for doing the behavior. Either I'm avoiding this shock, I'm getting away from something bad, or I'm getting this food, something good. They examine the effects of 2 drugs. And the way this is typically done is they establish this baseline, and actually this was done at Harvard, so on Tuesdays and Fridays were drug days. That's the Harvard regimen, with Thursdays serving as control. The animals were exposed to the experimental procedure 5 days a week, Monday through Friday. Tuesdays and Fridays are drug days, Thursday are control days. And so these doses are given a few days apart. The effects for amphetamine are probably the most spectacular because they show a reinforcement schedule effect that's pretty dramatic. This is the range of doses. The FI food, up and then down. FI shock, up and then down. FR food, cross and down. FR shock, cross and down. Doesn't matter what the consequence is. The effect depends essentially wholly on the schedule of reinforcement. Same true with chlorpromazine. Here you've got the fixed, the FIs actually are a little more sensitive here, go the other way. They go down together. FRs go down together as a function of dose of chlorpromazine. So the implication here is that it's the pattern of behavior that's crucial, not what's generating it. Now, it turns out this finding hasn't turned out to be entirely general. The major exception, though, that's in the literature is one of those scary ones. It's, the major exception is that if you compare food-reinforced responding to responding that is Greg here? Apparently reinforced by electric shock presentation. Anyway, you generate that pattern of behavior, very similar looking pattern of behavior, then the effects aren't the same. But there are several experiments to show that. But this kind of finding and the development of an analytic technique led to a lot of enthusiasm about the notion that what really counted was baseline rate of responding as a predictor of drug effects. That these had essentially comparable, the 2 fixed ratio schedules produced essentially identical rates, identical drug effects. Fixed interval schedules, essentially identical rates, identical drug effects. And the analytic technique, and I've never heard the story of how this was developed, but it's pretty clever, is the discovery that in a variety of circumstances, response rate is a really cool-looking predictor of drug effects. These are data from McKerney and Barrett on fixed interval schedule responding by monkeys. The way these analyses are done, you find some way to generate a variety of control rates. Now, it turns out the way this was most frequently done is by sort of a ruse. That you cheat. You do one fixed interval schedule, and you get as many response rates out of it as you want by chopping it up into a bunch of pieces. And so you say, I got a fixed interval 10-minute schedule. I can get you 20 response rates, one every 30 seconds. And that's what people tend to do. But there are other experiments where people use more conventional methods to get a variety of response rates. DRL schedule versus variable ratio schedule, those kinds of things. But with the fixed interval schedule, you get lots of points. Discovered if you expressed drug effect as a proportion of control, actually you don't have to express it as a proportion of control, but that became conventional, and plot that as a function of control rate on double logarithmic ordinates. These were here before the matching law, these double log coordinates, illustrating that it's a power function relationship between control rate and the rate observed under some effect of a drug. This would be a graph for a single dose. And it's actually it's a scatter plot. And then people fit lines to describe. And there are very orderly kinds of effects emerged from this kind of research that if the drug had no effect, all the points would go across the 100% line here. It would be flat. And as you increase the dose, what happens is the curve starts to tilt up, and it turns out some of the points, some of the high rates start going below the line control. So you got low rates going up and high rates going down. Same dose, same animal, same experiment, same session, making it look an awful lot like rate is a really important characteristic. Sort of lost in this was the notion that, hey, if all that matters is rate. That whole behavioral mechanism stuff, we can throw that out. Hardly need that. I just need the rate. It turns out it doesn't matter why they do it, what they're doing, how they're doing it. As long as they're doing it at some speed, you could predict the drug effect. And it turned out that didn't have as much generality. But it was thought for a while that this would account for lots of things. And that may be one of the reasons that we're not as far along as one might hope in trying to see if this behavioral mechanisms of action idea can even work. I promote it. We'll see if it gets somewhere. Another strategy for trying to illustrate that behavioral factors, how behavioral factors and drug effects might interact, and in fact this one sort of illustrates the idea of a behavioral mechanism of action, is also provided by Dews. This is an old classic. You always, in tutorial, you're supposed to use the classics, right? It's not like introductory psych books where all the references have to be 2 years old or younger, that there's actually some foundation of research. And what, actually I can just, let me put up the, I hope you can see this. It isn't reproduced beautifully. These are cumulative response records from Pigeon under multiple, up here under control conditions, a multiple fixed ratio 50, fixed interval 15-minute schedule. Sounds similar. These data were published, I think, in 1958. And so red key, it's FR50, blue key, it's FI15, and it produces these kinds of patterns on a cumulative response record. Then occasionally some sessions were preceded by an injection of chlorpromazine. And performance is screwed up to some extent. You notice the nice pauses seem to disappear. The animal just runs right through this. Boom. You know, the red happens. He just keeps going right on past, doesn't stop. So chlorpromazine, this dose of chlorpromazine is having a dramatic effect. And what Dews thought, well, geez, maybe what chlorpromazine does is makes it so, in essence, the animal can't see the stimuli anymore, or at least doesn't care to look at them, doesn't pay attention to the stimuli anymore, the red and blue keys. Well, what happens if we just take the red and blue keys out and see what kind of performance emerges? So, bingo. Doesn't look that much difference. So the notion that the action of chlorpromazine was somehow here to diminish attention to the discriminative stimuli is supported to some extent. Certainly not proved. But this is a kind of experiment suggesting that you can mimic a drug's effect by a behavioral manipulation. If you have some guess about what the drug might be doing to some behavioral process, you can, in fact, see what happens if you do that. And mimic, for example, lots of people think rate decreases are occasioned by, well, they're not hungry anymore. Well, we can certainly do that experiment. Does it produce the same pattern of effects? You just pre-feed them. Or, reduce deprivation if you have food-deprived subjects. Another, how am I doing here? I've got, what, 3 minutes? 2 minutes? I can go fast, as the students will tell you. I'll do this way. I won't describe, I'll describe the experiment as I describe the data. This is actually this is related to the kind of work that I do most of the time because it's a compelling way to show that behavioral factors are important. These are data from an experiment by Carlton and Wolgen, published in the early 1970s, where they had rats drinking milk, and they're looking at the anorexic effects of amphetamine. Amphetamine, you give the right dose of amphetamine, and you can get reduced intake of food. These rats are drinking milk, which they like to drink. There are 3 groups. These are group mean data. There are 3 groups. This is the saline group, the amphetamine saline, and the saline amphetamine. The order of these illustrates where injections of what took place with respect to the session. Amphetamine saline group got amphetamine before the session and a vehicle injection after each session. This group got the vehicle before the session and the amphetamine after the session. And this group is the ever-present control group. Never gets anything except saline, except on one day. And so what happens is the first day effect, this is the acute effect of a dose of amphetamine, of this dose of amphetamine. You see it reduces drinking in all these guys. So that's the give amphetamine once and then wait a while and start the real experiment. Here, these guys get amphetamine every day. Bing, bing, bing. You see they go back up into, this is the control level. They go back at maybe a little above the control level. This is an illustration probably of tolerance to the rate decreasing effects, or the milk drinking decreasing effects of amphetamine. The after group, they don't show anything. They're getting the drug after the session. Turns out giving drugs after sessions can produce effects it didn't here. And then on this day, they switch to this condition. Suddenly they're getting amphetamine before the session, and wham, they stop drinking. Now at this point, this group and this group has had exactly the same amount of exposure to amphetamine. These guys have had it 7 times post-session, and these guys have had it 7 times pre-session. These guys are all the way tolerant. These guys aren't tolerant at all. Something about being in the session while drugged has made the difference. The notion here is somehow the animal learns to compensate. Of course, nobody has any idea what exactly it's learning. There are people trying to figure out exactly how that learning might be characterized. But this has come to be called contingent tolerance. It's contingent on being able to practice whatever it is being disrupted while drugged. And so that's a clear illustration of behavioral contribution. This point out here, by the way, is not, like it is often in my data, some anomaly. But in fact, they gave the animals 3 milligrams per kilogram of amphetamine that day just to make sure they were still sensitive to the drug. Well, let me see if I can quickly go over this. And this is another contribution that behavioral pharmacology has potentially is examining drug effects for their implications about your interpretations of behavioral processes or processes of behavioral control. There are at least 3 ways this can be done. The example I frequently cite under option A is use drug-induced changes in behavior to tell you something about control mechanisms. Very quickly, this experiment by Vic Ladies, published back in the early 70s, used a fixed consecutive number procedure. I don't know how many people know what that is. With rats, you got 2 levers. And on one lever, you can get food. But to get food, you have to make some number of consecutive responses on the other lever. So this was an FCN 8 schedule. So 8 times on this lever, 8 or more. Press this lever, you get food. 7 over here, press here, tough luck, start over. And actually, the animals get quite good at this. The question is how they do it. That's one of the questions. Because perfectly correlated with number of responses is how much time, or at least well correlated, not perfectly, but highly correlated with number of responses, how long it takes them. So what are they doing? Are they timing this, or are they counting the responses in ordinary parlance? Well, how can you find that out? Well, it turns out that one of the drugs that Leite studies is haloperidol, which is Haldol, which had some interesting effects in that it produced no change in accuracy on this task, despite the fact that it slowed response rate tremendously. So that points to, that's at least some evidence in support of the notion that count is what counts. That if it doesn't matter whether it takes you 2 seconds to make the 8 or 25 seconds to make the 8, you still choose the right lever at the right time, suggests that number or some really fancy interaction between timing and count is going on. But it implicates count as a controlling variable. There's a lot of activity these days. In fact, probably the majority of behavioral pharmacology these days is aimed at this with drugs serving stimulus functions, either reinforcing or discriminative or eliciting, punitive. One of the things that does for an interpretation of behavioral processes is it extends the generality of what act as effective consequences, what act as effective stimuli. The fun things about them serving as discriminative stimuli is that you can argue that you're establishing control by private events for people who are interested in those kinds of issues. But you have control over administration of the drug that produces presumably some internal set of stimuli which serve a discriminative function. And you are, fortunately, you're the public that has guaranteed that that's in there. And then by far the most common is the use of drugs as added stimuli. You have some behavioral preparation going, you administer a drug, and sometimes the drug effects can tell you things about the nature of behavioral control. Instead of showing you the graph I brought, too bad, Steve, was some data with you. But anyway, I'll show you the data are beautiful. And so I just won't describe this experiment. I'll describe another one that was reported several years ago that might be of interest if there are any conditional stimulus control people in the audience by Newland and Marc, where they did a very simple experiment. They trained up some matching to sample in pigeons and then looked at the effects of chlorpromazine. And sure enough, as you might guess, as you increase the dose of chlorpromazine, accuracy on the match to sample task was impaired. But they chose to examine accuracy separately by stimulus configuration. You know, so you got green on the center and green on the right and red on the left. That's one. Peck the green. Get green in the center, green on the left, red on the right, peck green. Most people aggregate those together. Well, what happens if you look at the accuracy separately? The curves aren't the same. That in fact, the different arrangements were controlling behavior in different ways, only revealed when the drug was given. And I think lots of people have gone to good lengths with other methods in behavioral analysis to show that, in fact, particular stimulus arrangements, for pigeons at least, can be controlling factors that they're not in there, matching to sample like all of us identity people would do. And these are some nice data that Steve helped produce, illustrating that positive and negative punishment aren't influenced in exactly the same way by, even though everything else is pretty much similar, aren't influenced in the same way by pentobarbital. And my time is up. Thanks. By the way, the answer was E. Nice question. I planted her. Behavioral factors sometimes. That's actually something I've been working on for quite a while. There are circumstances, at least with respect to cocaine, which in many circumstances, repeated exposure to cocaine will produce sensitization to its effects. Typically not with operant performance, which is kind of that sort of standard operant condition performance. The more common result is tolerance if you give the drug repeatedly. But that can depend on behavioral factors like schedule parameter and stuff like that. It's they're psychostimulants. You've got a better chance with psychostimulants to produce sensitization than you do with a lot of other drugs like morphine. Pretty hard to produce sensitization with morphine, although you can get it if you work hard at it. But that's one of the things that makes life interesting. Pharmacologists can, in many circumstances, sort of tell you what's happening in parallel when sensitization occurs. You got receptor upregulation or something like that, and so you got more receptors going off when the drug's in there. But the mechanism, even the physical mechanisms by which that occurs aren't entirely clear. That has to be a change in gene expression, probably, if you got more receptors. Yeah, Marc.
Someone who's completely ignorant about pharmacology, could you characterize briefly the current impact of pharmacology on psychopharmacology? What's the relevance? How many people are doing it? Are the others listening?
Well, here's my perception of that. I don't have any real data. Is that that psychopharmacology acknowledges behavioral pharmacology in a couple of ways as mainstream pharmacology. One is drug self-administration as a screen for potential abuse liability and as a model for trying to develop drug abuse treatment procedures. Those of you who heard Bob Schuster talk this morning, he gave an example of how that's developed. Drug discrimination procedures, drugs as SDs, has become almost a standard whole animal assay for examining drug receptor interactions. I'm not sure where that's going. Actually, I've always been a skeptic of that, and in the last 10 years, more data have come out, I think, to make people who are in that business a little more skeptical. There's some miracles have occurred in that. That's the only way I can describe them. And you have to pay attention to miracles. That if you train an animal to discriminate, say, pentobarbital from from saline. So on some days you get pentobarbital, you have to press the right lever to get food. Other days you get saline, you got to press the left lever to get food. So it's a everyday alternating multiple schedule, essentially. And animals learn this. They get good at it. And then you give, some other drug, like alcohol. Doesn't look anything like pentobarbital. Well, what does the animal call it? Calls it pentobarbital. And then you discover, wait, we discover later that alcohol and pentobarbital share a common receptor. Alcohol goes all over the place, but son of a gun, it turns out that things that act at similar receptors, you can pick them up in this behavioral assay. And that's a miracle, because when you put a drug in there, it may go to some receptor, but if that receptor does anything, stuff goes off everywhere. You do your fMRI, lots of places in the brain are lighting up, not just little pieces where this receptor is. So what is it about this thing? Well, what's happening, of course, people have now discovered that those stimuli are quite multidimensional, and so and are trying to actually either take advantage of that or cope with it. But that's certainly very. The major journal, one way to look at this, the major journal in pharmacology is the Journal of Pharmacology and Experimental Therapeutics. That's the big stud journal, and it has lots of drug discrimination, and it has lots of drug self-administration stuff, and some other kinds of behavioral pharmacology, as well as another more standard psychopharmacology. And depends on what you include in behavioral pharmacology. People are interested in inventing drug screens that are simple and fast and efficient. So you have, the elevated plus maze, where you don't have to train the animal to do anything. You just stick them on it, stick the drug in them, and see where they go. Those kinds of things. If you count that as behavioral pharmacology, there's lots of that stuff. And then, as long as I'm in a friendly audience, then there's this horrible conditioned place preference procedure, which has 7,000 potential interpretations. But it's very popular because it's really easy.